Good afternoon, dear delegates, and welcome back to the session 2, scientific session 2 of Day-9, Series 3 online FDP, sponsored by AICTE and organized by Roland Institute of Pharmaceutical Sciences. Dear participants, we are having our next eminent resource person, Dr. N. Srinivas sir, with us. Before inviting sir for his presentation, it is my privilege to introduce Dr. Nami Srinivas sir to all of us. Dr. Srinivas sir, Srinivas is a senior lecturer in pharmacology and academic course advisor medical sciences at the Discipline of Medical Sciences, School of Science, Western Sydney University, Australia. Dr. Nami holds a doctorate in pharmaceutical sciences specializing in pharmacology from Andhra University, India. Before joining Australia, he worked at the Faculty of Pharmacy, University of Sydney, after gaining postdoctoral experience as DAAD Fellow at the University of Division, Germany, and at the University of Manitoba, Canada. Dr. Nami also worked as a head of pharmacology in an industry where he was instrumental in establishing a new pharmacological research laboratory. Dr. Nami has attracted competitive research funds from various national bodies like ARC, AISRF, NICM, and international body like India UGC and CSIR in Germany, DAD, as well as institutional and industrial bodies. Sir is having one international patent and has published over 90 scientific papers, 10 editorials, and 3 book chapters. Doctor Nami's research has been cited so far in over 3,000 publications with an H-index of 30. Sir has supervised 8 PhDs, 8 masters, and 15 honors projects to completion. Dr. Nami has significant professional outreach through his contribution in academic and scholarly activities. He is serving as secretary for the Australian DAAD Alumni Association. He is an international expert reviewer of a number of national and international research grant bodies. He sits on the editorial and review boards of a number of reputed biomedical journals. He is also frequently invited to guest and edit different journal special issues and to edit and contribute to books and other book chapters. Sir has been awarded a number of fellowships and awards, including the prestigious DAAD Fellowship, Germany, Sescu Fellow Postdoctoral Fellowship, Australia, and CSIR, India. In 2018, Sir has been awarded Research Excellence Award by the School of Science and Health, Western Sydney University for his outstanding contribution in the field of scientific research. With this brief introduction, may I now invite Dr. Srinivas Nami sir on board to kindly join us and to deliver his talk.
Thank you, Dr. Rajaram, for your kind introduction.
Thank you very much.
So before I start sharing my screen, good afternoon, everyone. I would like to thank my long-term friend, Professor B.V. Ravikumar, who is the principal at Roland Institute of Pharmaceutical Sciences, for giving me this opportunity to present my research and share all my background and future ideas with you. I welcome all the various faculty members coming from various pharmacy colleges around India. And I thank you everyone for attending this presentation. Shall I start sharing my screen?
Yes, please.
Right. May I know if you're able to see the screen?
You make it full screen, Nami.
Full screen, okay. Yes. Is it full screen now?
No,
At your no?
no.
It is full screen on my screen.
But it is not here.
Uh
Actually, you have to go. You stop sharing once.
huh.
You go to share. Share option is there, na?
Share
Uh,
option.
After that, share screen is there. Share option
So what you see on your screen now?
on my screen, nothing.
Okay.
You,
So yeah. So I went to share, and then I have
video
selected
file
share
and the
screen.
share screen. Two options are there.
Yeah. And then once I
Share
click
screen.
the share screen, um,
Then you'll see some screen sharing tips. Some screen sharing tips.
so once I click on share screen, I have three options there. Screen sharing is easiest in
No,
with
no,
group.
no. Those are only tips.
Yes, yes. So I click on share screen at the
Uh,
bottom.
uh,
Then I have view entire screen application window,
uh.
and then Chrome tab,
Entire screen.
entire screen. Okay,
It
and
is
then
highlighted, and click the bottom portion.
um,
Entire screen.
right.
Then you highlight that one. Whatever is. Uh,
Okay. What you see
then
now?
you click. Yeah,
Are you able to see now?
yeah. But
Full
it is
screen
not
or
not full
how about
screen.
now?
Now it is full screen. Now it is full screen.
Okay, good, good. Okay.
Mm.
I think we had the same issue last time as well.
Yeah.
Right.
You can you see Nami below your name, one horizontal
Uh-huh.
bar is there. There you click
Okay.
hide.
Right.
Okay.
Okay. All good now?
Yeah, absolutely perfect.
Okay. Right. Good afternoon, everyone. My name is Srinivas Nammi. I'm a faculty member at Western Sydney University School of Science and Health. Um, I hope you attended my one of my previous lectures as well a few months ago. So I was originally from India, myself along with the Professor B.V.V. Ravikumar. We both studied at the same university, Andhra University in India. And then I did some postdoc in Germany, Canada. Eventually, I moved to Australia to the University of Sydney and then to Western Sydney University. So, what I'm going to share today is my journey with one of my research areas on alcohol and metabolic disorders, coupled with non alcoholic fatty liver disease, and to some extent into alcoholic fatty liver disease as well. So, this is my research over the years for nearly last 20 years. If you can see here, we started this alcohol research way back in 2005 2006 when I was doing my postdoc in Canada. I was fascinated into this research at that point to look at what happens if pregnant women take alcohol during their pregnancy, whether that in utero alcohol consumption has any um, bad effects on offspring in the immediate life or in their later life. So, on those lines, we took that project and we saw that when pregnant rats were given alcohol in the sense heavy alcohol to those rats, we found that some of the signaling pathways which promote obesity and type 2 diabetes in the offspring, they're regulated. For example, we noticed that 11 beta HSD1 and also hexose 6 phosphate dehydrogenase in both liver as well as in adipose tissues. So they were regulated in the direction to induce obesity and type 2 diabetes in the offspring. So that was like alcohol toxicity in the that is created in the fetus, and then uh, a program that has been developed in the fetus to develop type 2 diabetes in the offspring in the later life. Later, we also looked at what um, if those pregnant women consume light and moderate alcohol, whether they also um, uh, promote any type 2 diabetes and obesity in the offspring or not. Unfortunately, we couldn't actually try that part within that project duration. But later, what we tried was to have that light and moderate alcohol direct consumption, not in pregnancy, but in adulthood. For example, when um, light and moderate alcohol was consumed, whether that has any um, deleterious effects on the biological system as compared with heavy alcohol consumption. We all know that heavy alcohol consumption can induce fatty liver disease, and what we term that one as alcoholic fatty liver disease. And um, most of us also know about the pathophysiology of alcoholic fatty liver disease and the various signaling pathways established and confirmed within the development of fatty liver disease. But we were trying to look at that point around 2007 or 8. During that time, we were looking at light and moderate alcohol, whether it can actually increase insulin sensitivity, and as a result of that, whether it can promote any beneficial effects in order to prevent or delay the development of type 2 diabetes. So on those lines, we found that for the first time that light and moderate alcohol, it upregulates AMPK alpha, which is one of the master regulators for both glucose as well as for lipid metabolism. And that was one of our interest in our research areas, to look at AMPK expression and also the upstream as well as downstream targets of AMPK. So in that project, we looked at AMPK along with some other downstream molecules of AMPK, plus one of the molecules that induces lipid biosynthesis, SREBP. So we found that light and moderate alcohol downregulates SREBP and upregulates AMPK in healthy rats. So that indicates that healthy individuals, when they consume light and moderate alcohol in moderation, okay, again, so I'm going to talk in my lecture what's the threshold of alcohol consumption and the frequency of alcohol consumption, and how do we define light, moderate, and heavy alcohol. So when individuals consume light and moderate alcohol within the limits, they have enhanced insulin sensitivity. At the same time, upregulation of AMPK, which allows glucose metabolism and lipid metabolism, fatty acid oxidation, and at the same time reduces SREBP. So de novo lipogenesis is inhibited. So that was a positive outcome at that point. And then we further um, proceeded to see what if um, light and moderate alcohol is taken along with high fat diet, for example, because in general population we see that people consume high fat containing foods all the time, and at the same time they consume maybe alcohol, and sometimes people take alcohol in moderation, and for their benefit they always say that a glass of wine a day keeps your doctor away, same like an apple a day keeps your doctor away. um, but again, we need to see whether those commercial beverages containing light to moderate alcohol, what the general um, saying of the general population that the light and moderate alcohol is good for health. So we looked at some of those commercial formulations as well, which was not discussed in this particular presentation, but we came to know that some of the general commercial formulations do contain a lot of polyphenols, and some of the beneficial effects can also be attributed to those polyphenols. So we decided not to take any commercial formulations for this particular research project. Instead, we wanted to take plain ethyl alcohol at different concentrations prepared in the laboratory, and then give that to our experimental animals at light and moderate concentrations to see whether plain ethanol itself has got any positive or negative outcomes in those animals. So, so my entire presentations will be surrounding those ideas, what I just mentioned. So just to take you back to the evolution of metabolic syndrome, we have been hearing about many definitions about metabolic syndrome over many generations. So for the first time, for example, in 1923, Kailin described metabolic syndrome as a cluster of hypertension, gout, and hyperglycemia. Later on, it again came into existence in 1988 when Gerald Reaven described metabolic syndrome as syndrome X, constituting hypertension, hyperglycemia, glucose intolerance, elevated triglycerides, and low HDL cholesterol. So that was the definition given in 80s. And again, in 90s, WHO refined it further, saying that metabolic syndrome rather than syndrome X, with a cluster of hypertension, again, low HDL, high triglycerides, insulin resistance, glucose intolerance, type 2 diabetes, and also added few more things like high waist to hip ratio, and also presence of microalbuminuria. And during the last few decades, we also went on further converting that or changing that definition from metabolic syndrome to cardiometabolic syndrome. In recent years, we started hearing about cardiometabolic syndrome because that definition by WHO in 1998 was not enough, because we started seeing cardiovascular dysfunctions along with all the various manifestations what we have seen here. And in that cardiometabolic syndrome, non-alcoholic fatty liver disease is also added as one of the hepatic manifestations of the metabolic syndrome, because people started to develop metabolic syndrome, also develop fatty liver disease. They start with steatosis, and then slowly it progresses to steatohepatitis. So there will be some inflammation along with accumulation of lipids. And then if not address the problem at that stage, it can also lead to liver cirrhosis and liver cancer. And we also see that metabolic syndrome or pre-diabetes, we can also term as a pre-diabetes, as we know. So that is also strongly associated with non-alcoholic fatty liver disease, and also linked with type 2 diabetes, because around 70% or more than that of the type 2 diabetic patients, they still have non-alcoholic fatty liver disease. And like I said, it develops the steatosis, also termed as NAFLD, and non-alcoholic fatty liver disease, and slowly it progresses to NASH, where the pro-inflammatory molecules play a crucial role, and then it develops into fibrosis, and then to cirrhosis and HCC, hepatocellular carcinoma. And it not only in adults, we also see that childhood obesity and accumulation of fatty molecules or lipid droplets within the liver in youngsters as well. So the cardiometabolic syndrome, like what I was mentioning before, constitutes various factors, like they have cardiovascular dysfunction, heart disease, dyslipidemia, hypertension, and eventually develop into type 2 diabetes. And also those patients have NAFL, non-alcoholic fatty liver disease, plus dysfunction of the brain, dementia, and also cancer and polycystic ovarian syndrome also seen in females. So on one side, we know about alcoholic fatty liver disease, but on the other hand, we also know about non-alcoholic fatty liver disease. But when we coined this project, when we designed this project, so we need to make it clear for our reviewers where we call it as non-alcoholic fatty liver disease and why we are actually using light and moderate alcohol within fatty liver disease to look at the outcomes of moderate alcohol consumption. So if we look at the guidelines of American Association for the Studies in Liver Diseases, so that's a society which provides guidelines and defines non-alcoholic fatty liver disease by taking some criteria into consideration. So what they said was they have excluded the people taking more than 14 drinks per week for women and more than 21 drinks per week for men. So they have excluded those individuals. And also, it was noted that alcohol intake as low as 20 grams daily in women and 30 grams in men may also be sufficient enough to cause alcoholic fatty liver disease. So in some guidelines, they say that 20 grams of alcohol is considered as moderate. In some guidelines, it is also shown that 20 grams of alcohol in women, that we need to make that note, that is sufficient enough to cause alcoholic liver disease. So there may be some variations, individual variations as well. So whether we should consider ethanol consumption in definition of non-alcoholic fatty liver disease, same like what we did with chronic hepatitis C or not, it's not very clear. But at the moment, for non-alcoholic fatty liver disease, we are not giving any alcohol to induce that. We try to do with other modes of development of the disease. So patients with non-alcoholic fatty liver disease should not consume heavy amounts of alcohol. So that's what ASL guidelines says. But again, it's open for debate across many bodies as well. So what we see here is the central point is the development of insulin resistance in the development of fatty liver disease. So insulin resistance, as we know, that enhances SRABP expression as well as carbohydrate regulatory binding protein. They both cause triglyceride accumulation, and also we see that in the adipose tissue due to insulin resistance, we started to see decline in adiponectin and upregulation of leptin, and also we see increased fatty acids. So that leads to accumulation of lipid droplets in the liver cells. So when it comes to the etiology and pathogenesis of fatty liver disease, I'm just using fatty liver disease because we share some of the etiology and pathogenesis in both non-alcoholic and alcoholic fatty liver disease. So if you look at this slide, high calorie intake, sometimes dietary fat as well. So we all know that induces obesity through various mechanisms, downregulation of adiponectin, which again cause decreased fatty acid oxidation, insulin resistance, cause increase in lipogenesis, de novo lipogenesis, and also increased influx of fatty acids to the liver and accumulation of fat in the liver. And we also share similar pathways with alcoholic fatty liver disease as well. So when we look at this slide, this shows alcohol-induced fatty liver disease. For example, high doses, I call it as heavy alcohol consumption rather than high dose. So heavy alcohol through its metabolites, for example, we all know that ethanol undergoes metabolic process through alcohol dehydrogenase pathway and also the cytochrome P450 pathway as well. Specifically, the 2E1 isozyme is involved in ethanol metabolism. So through those metabolic pathways, we know that acetaldehyde is formed as a byproduct and eventually is converted to acetate. And also at the same time, oxygen species, reactive oxygen species are developed in both pathways. There may be some differences in the magnitude of reactive oxygen species in those two respective pathways, but we see reactive oxygen species which eventually cause increase in lipid accumulation, and also the pro-inflammatory cytokines, interleukins and TNF alpha. So they all play a crucial role in steatohepatitis. And we see here at the top the high fat containing cafeteria diet we are all used to nowadays. So that causes both glucose toxicity as well as lipid toxicity. So increase in glucose and increase in lipids causes glucose intolerance, insulin resistance, and accumulation of free fatty acids in the liver cells, and eventually develop into in fibrosis and steatohepatitis. So what we see common with alcoholic and non-alcoholic fatty liver disease is, so this is one of the central pathways for the development of fatty liver disease. When individuals consume heavy alcohol, the master regulator, which is AMPK, AMP activated protein kinase, so that is known to be downregulated, and as a result of that, the downstream targets of AMPK are regulated. For example, ACC, acetyl-CoA carboxylase activity is enhanced because AMPK inhibits this. When AMPK is downregulated, you see increase in ACC, and that in turn increases methylmalonyl-CoA, which reduces CPT1 activity in the mitochondria, that reduces fatty acid oxidation. And at the same time, AMPK increases SRABP, downregulation of AMPK increases SRABP, that cause regulation of various lipogenic enzyme expression. They're all upregulated, that causes increased de novo lipogenesis. And also you can see here methylmalonyl-CoA decarboxylase is downregulated when AMPK is downregulated, and that this particular enzyme metabolizes methylmalonyl-CoA. So when this is downregulated, so indirectly methylmalonyl-CoA is enhanced, and that also contributes for the fatty acid synthesis. So that is known for heavy alcohol consumption. All these pathways were well established, so it's all confirmed. With high fat diet consumption as well, we notice that similar mechanisms have seen. For example, with AMPK, high fat diet is known to downregulate AMPK, and high fat diet is also known to downregulate SIRT protein, which is sometimes it is working at the same level of AMPK, sometimes at a level higher or lower than AMPK. But majority of the reports, they show that SIRT and AMPK, they try to regulate each other. Downregulation of AMPK, again, whatever we have seen here, you can see all those pathways in similar directions. Methylmalonyl-CoA decarboxylase is downregulated, ACC is upregulated, and eventually you see increased fat accumulation in the liver, leads to NAFL. So these pathways with high fat diet is also well established, so we don't have to prove them now. They are in parallel, well established along with heavy alcohol consumption. So we know how the alcoholic and non-alcoholic fatty liver disease development process happen, the pathogenic pathways. But what we are hypothesizing is that low and moderate alcohol, we took 6 and 12% alcohol to match with the percentage of alcohol that is seen in beer, for example, and the percentage of alcohol that is seen in wines. The average percentage is 12% in wines. So we're trying to look at whether light and moderate alcohol can act on any of these pathways. And we also know recently that liver kinase B1 is also playing a crucial role on top of AMPK and SIRT. And little is known about this particular protein. So we want to see whether light and moderate alcohol is regulating the pathway through AMPK or through one of the downstream targets or even the upstream target, which is LKB1. So that was our idea to start the whole project. And this project was conducted for the last 4 or 5 years by a couple of my PhD students. And a little bit more before I actually start presenting what we have done is the AMPK. We know that AMPK is a master regulator which regulates both glucose as well as lipid metabolism. And there are various analogs of AMPK are available. So we have some ligands which can stimulate AMPK, and at the same time we also have one or two inhibitors of AMPK which can be used in research. So what I want to say here is AMPK, like I said, it can influence its immediate downstream target, which is ACC, and as a result, the CTP1 activity can be altered to show the positive effects. It can increase fatty acid oxidation. At the same time, the de novo lipogenesis can also be inhibited. So, like I said, different countries and different regulatory bodies have got different definitions for the threshold of alcohol consumption, what is called as light and what is called as moderate heavy binge. So what I'm presenting here is from the NHMRC, National Health and Medical Research Council of Australia. So they have developed their own guidelines. And because we conducted all this research in Australia, we had to obey the NHMRC guidelines. We had no other choice. So what we did here was, following the guidelines, we defined that one standard drink. It's not we defined, NHMRC guidelines define that one standard drink is equivalent to 10 grams of alcohol, and consumption of one standard drink per day is considered as light alcohol drinking. And that is by a person with an average body weight of 60 kilograms. In the same way, 2 standard drinks per day is defined as moderate. More than 2 standard drinks per day is defined as heavy alcohol. And people also consume continuously. They continuously they drink more than 5 standard drinks on a single occasion. That's classified as binge alcohol drinking. So we haven't done any comparative studies on this one in our projects. So we were interested on light, moderate, and also in some of our experiments we also looked at heavy alcohol consumption as well in parallel to see in our experiments whether we get any differential effects or not. So, ladies and gentlemen, so you can remember that one standard drink per day is light, 2 standard drinks, which is equivalent to 20 grams. So 10 grams is light, 20 grams is moderate, more than 20 is heavy alcohol. So, like I mentioned earlier, this is one of our motivating pathways in order to conduct the entire project, because we hypothesize that people consume low and moderate alcohol through the commercially available alcohol beverages. If they get any positive effects, it's not due to the other molecules present, not because of polyphenols or not because of any other carbohydrate molecules present within those beverages, but we strongly feel that ethanol itself, through one of its metabolites, can show some of the positive effects. Of course, it can show its negative effects. That's what we see with heavy alcohol consumption, and that's well established as well. But we say that light and moderate alcohol can follow one of these pathways to show those beneficial effects. And what is that pathway is, we strongly believe that light and moderate alcohol is following the alcohol dehydrogenase pathway to produce acetaldehyde, and then with the help of aldehyde dehydrogenase, it's converted to acetate. And acetate may have some positive effects. I'll tell you later how it has positive effects. But at the same time, the cytochrome P450 pathway contributes to a tremendous increase in reactive oxygen species. And that is one of the known pathways for the development of alcoholic fatty liver disease. And one more important point is, we all know that ethanol can induce liver microsomal enzyme. It's an enzyme inducer. So what I want to say here is, at low concentrations, it follows the alcohol dehydrogenase pathway, whereas when individuals consume heavy alcohol, the CYP2E1 is induced, and as more CYP2E1 is induced, then the alcohol metabolic pathway is diverted towards the CYP pathway rather than ADH pathway. So that was our argument at the beginning, but we partially proved that, and we are still doing on those lines. So the central hypothesis for our entire research was long-term consumption of low and moderate alcohol protects the liver from the development of non-alcoholic fatty liver disease. So in order to prove that, we developed methods along the line. So to look at all the observations, what we can see in animal studies, look at all the serum biochemical parameters as well as the tissue parameters, and also conduct the histopathological studies in tissues, and also look at some possible mechanisms along the AMPK pathway where we are interested in, to see whether light and moderate alcohol regulate one of these molecules, and to look at the specific mechanisms, whether AMPK is important or LKB is important or SIRT is important, if at all we see any positive effects. So that was one of our objectives. And also we wanted to look at, we all know that the animal regulatory bodies, they try to limit the usage of animals. So we have those ethical regulations as well everywhere around the globe. So we wanted to find out some of the mechanisms in culture models rather than using more and more animals for the mechanistic studies. So we wanted to develop parallel studies as well, using HepG2 cells, HeLa cells, and then try to demonstrate those mechanisms in those cell lines by different means, using pharmacological inhibitors or genetic manipulations, etc. So that was our objectives, and we started to conduct the study with different phases, following all the objectives what we had. So in stage 1, our researchers conducted an observational study. In similar studies have been conducted around the globe. I hope this is very clear for you as well. So we conducted studies in high-fat induced, high-fat treated animals. It is a protective study rather than a curative study. So from day 1 onwards, we started treating the animals with high fat, and we started giving the alcohol at different concentrations. So low dose of alcohol, 1 gram per kg per day. Moderate alcohol is 2 grams per kg per day. So we prepared the 12% and 6% alcohols on daily basis freshly. And for the heavy alcohol consumption, we use 36% alcohol. And based on the standard definitions, we converted the human doses back to the animal doses, and we dosed all these animals for up to 15 weeks. So this study was conducted for 15 weeks with the different groups ran in parallel. We also have the high fat along with heavy alcohol consumption on a separate group just for comparison purposes, but we were interested on light and moderate alcohol effects on non-alcoholic fatty liver disease. So in stage 1, along with along with animal studies with alcohol, our researchers have also conducted parallel studies on cell culture models, because towards the end, we wanted to prove some of the mechanisms in cell culture models, and we wanted to run the experiments in parallel. So the HepG2 cell model was used. The oleic acid and palmitic acid combination at the concentrations given, it was used to induce the lipid accumulation in the cell culture models. And when the treatments were conducted, we measured the lipid parameters. We measured the triglycerides as well as cholesterol in those cell lines. So with the animal studies, we came to know that there was no significant increase in body weights as compared with the negative controls. So we have the negative control, which is healthy negative control, as well as the disease negative control, and we can see the low, moderate, and heavy alcohol consumption. Even though there was a tendency to gain weight in heavy alcohol consumption group, but there was no statistical significance. And you can also see here these bars where the comparison of the body weights at the start of the experiments at 0 week and after 15 weeks. So you can clearly see that if you compare the black bars, so compared with the disease negative control, so both the low alcohol as well as moderate alcohol, there was not much variation in the body weights. However, we have seen some variations with some of the biochemical parameters. But if you see here, the disease model was working fine. We can see that there's an increase in food consumption, or a decrease in food consumption in the high-fat diet. And you can also see, even though there is non-significant increase in body weight, but there is a tendency to increase the body weight regardless of decrease in food consumption. And the decrease in food consumption was also seen in low, moderate, and heavy alcohol. So one of the reasons for that is in these groups we can attribute that to the alcohol impalatability, whereas with high-fat diet, the food consumption was reduced, so they didn't like the fatty food. So the total energy intake, you can also see the total energy intake is increased, even though it's not significant here. Liver weights, you can see the liver weights is increased, so there's some accumulation of lipids happening in the high fat, as well as even though it's not significant here, you can see, so there's an increase in liver weight, and liver weight to body weight ratio as liver index, we also have similar results here. So when it comes to the lipid levels in the serum, we notice that triglyceride levels were significantly increased along with cholesterol in the high fat negative control. And you can also see here with low, moderate, I'm not talking too much on the high fat, high alcohol at the moment. So you can see here with low and moderate, you still see an increase in the levels, even though they didn't achieve significance. HDL cholesterol was significantly lowered with high fat and significantly increased as compared with this one here, disease control. So you can see significant increase in both low and moderate. LDL 300, and then you have 304, 226. Similarly, fatty acids. So we noticed significance in some molecules, whereas we didn't see significance even though we see the trends. So that's one of the reasons is that we didn't have enough rats at that point. In some groups, we only had 4 animals. In some groups, we had up to 6 animals, because we noticed that some animals died during the process. When it comes to the glucose and insulin measurements, you can see here there's a significant increase in glucose with high fat consumption, and at the same time significant increase in insulin as well with high fat. So there is insulin resistance. With low alcohol, you can see it came down to 359 from 372 and further lowered. Insulin levels again not significantly reduced. The AST and ALT was also measured. You can see here it was significantly reduced with moderate alcohol, but no such significance noticed with low alcohol. So there are some slight differences with the low and moderate alcohol effects. We looked at the triglycerides and cholesterol in the liver as well, because that was important for us to see whether there is lipid accumulation or not in the liver. So with high fat treatment, we noticed a significant increase in triglycerides and total cholesterol as compared with healthy rats. With low alcohol and moderate alcohol, so you see here there is a tendency to lower the triglyceride levels, but not significant enough. Cholesterol, with moderate alcohol consumption, there is a significant reduction. With low alcohol, we didn't observe any change. In fact, a slight increase here. So you know that we ran the cell line experiments in parallel, where we also measured the cholesterol and triglycerides in the cells after treatment with different concentrations of alcohol. You can see here the triglyceride concentrations, again, significant increase with the fatty acid treatments in line with the high fat treatment in the animals, whereas the moderate alcohol and low alcohol, they didn't produce that much significant reduction here in both cholesterol and triglycerides. And we also did, because it's cell lines, it's easy to run various groups. So we did treatments in normal cell lines as well, not only with fatty acid treatments. So in healthy cells plus low alcohol and moderate alcohol, you can see here. So compared with this, there is a slight increase in triglycerides, and here not much. And similarly with cholesterol, you see in normal case with low alcohol, you have a slight increase in total cholesterol and also a slight increase in cholesterol in the moderate animals as well. So we wanted to find out what actually caused the increase in triglycerides and cholesterol with low and moderate alcohol. That was something different as compared to our previous results. So then we went on to look at the histology, where we looked at the basic morphology with H&E staining, and also we looked at the oil red staining to look at the neutral lipid accumulation. So these slides are taken at 20x. So I try to fit all the slides in one screen. So where we can also see the central slides here, A and F, they are the slides from the healthy animals, from the normal control. and then the B is high fat, C is low alcohol, D is moderate alcohol, E is heavy alcohol, and the same sequence that is followed here. So this is more like a qualitative assessment that is presented here. So we notice that there is some improvement with light and moderate alcohol as compared with heavy alcohol consumption. So then we went on to see what mechanisms are actually working on in order to protect the development of lipid accumulation in those rats. So we looked at AMPK alpha and also the upstream and downstream targets of AMPK. And ladies and gentlemen, please bear with me. I'm going to show you lots of Western blot results. So you may be getting bored about these Western blots now. So I will be showing lots of screens of Western blots. So we see here, this was in from liver tissues in coming from these experimental rats. So you see here there is a downregulation of AMPK as well as the phosphorylated AMPK. So we looked at both phosphorylated and unphosphorylated protein. So downregulation of AMPK was seen with high fat, and that was already proven in literature. So that's not a novel finding for us. But what we see here is with low and moderate alcohol, as compared with high fat control, we started to see an increase in AMPK. And again, that was for comparison purpose, high alcohol is reducing AMPK. And this was also reported in literature. So our novel findings were these two. And similarly, we have phospho-AMPK, increased phosphorylation. So both protein, both expression of AMPK as well as its phosphorylation is enhanced with light and moderate alcohol. In parallel, in the cell line studies, after treating the cells, HepG2 cells with different concentrations of alcohol, we looked at AMPK and phospho-AMPK. You can see here with low alcohol, the AMPK protein was not altered, whereas with moderate alcohol, you see an increased consistent with the liver studies, and also with phospho-AMPK. With moderate alcohol, we see consistent with this and also consistent with our animal studies. But with low alcohol, we didn't see a significant change in both AMPK as well as in phospho-AMPK. So one of the reasons for that one is possibly the alcohol concentration was not enough, or the number of samples were not enough. Then we looked at the LKB, liver kinase B1, which is considered as an upstream kinase of AMPK, as we have seen in one of the slides there. So again, LKB and its phosphorylated protein is measured using Western blot studies. We noticed downregulation of LKB and phosphorylated LKB with high fat diet treatment, and that's also it's reported in some papers, but it's still in infancy stage. So high fat diet consumption in LKB is still lot of scope to do research on those lines. But what we noticed here is low and moderate alcohol consumption. You can see here a slight increase, but no significance with the low alcohol on the LKB protein. And also it's on the phospho-LKB. But you see here the moderate alcohol enhances the phospho-LKB1. So it enhances the phosphorylation process of LKB1 even though there is no change in expression of LKB1. Whereas with low alcohol, you see both increase expression of LKB1 as well as increase in phosphor. However, there is no significance. So that's something we need to further study to see what actually caused those differential effects. In one case, we see increase, in one case, no change at all. Then with the cell line studies, when we looked at LKB and phospho-LKB, again, with the low concentrations, again, consistent with our animal results, there was no significance achieved, but there is a tendency to go up. Whereas with moderate alcohol, you can see there is a significance with LKB protein expression significantly increased, and also its phosphorylation. So as compared with our previous one, so we see here increased phosphorylation. So with animal studies, we didn't see much change, possibly because of less number of animals in that group. So we need to further address that. So then we looked at other molecule in line with AMPK, which is SIRT1. So sirtins are one of the molecules which also regulate lipid levels in liver cells. So what we see here is in the rats, in the liver, low alcohol is increased, but no significance. And also the moderate alcohol produced increased SIRT without any significance. But there is a tendency, you can see. So we need to, this is marginally achieving that 0.08, whereas here we need to look at considering more animals in future. In the cell lines, we notice that both low and moderate produced a significant increase in SIRT1. So we are hoping that in animals also in future, when we conduct with more animals, we are going to achieve that significance. So then we looked at the downstream targets of AMPK. ACC is one of the downstream targets. We looked at both the ACC protein expression and its phosphorylation. You can clearly see here with high-fat diet, the ACC protein expression is increased because we know that AMPK is downregulated, so ACC is increased, and that's consistent with literature findings. But the phosphorylation is inhibited. So that's something, I don't say it's novel, but that's something strange as reported from literature. With low and moderate alcohol, we see that the ACC expression is reduced with both low and moderate, even though there is no statistical significance. But here with the phosphorylated protein, we noticed an increased. But whether this is correct or whether this is correct, we need to further address. Whereas here, we still need to do more studies to get that significance. This is completely strange as compared to literature findings. So then we looked at the ACC again in parallel in the cell culture models, where we see increased ACC, and then you can see with the light and moderate alcohol. With moderate alcohol, we see a significant reduction in ACC. The phospho ACC with moderate alcohol, a significant increase in ACC as compared with free fatty acid control cells. Then we looked at one of the molecules for de novo lipogenesis, SREBP1, in both liver as well as in liver cells. With high fat treatment, SRABP1 expression is increased significantly. With low alcohol, SRABP1 expression is reduced, whereas with moderate alcohol, in fact, it is increased, even though there is no significance. In the cell culture models, with moderate alcohol, with both moderate as well as with low alcohol, we see no significance here. In fact, a slight increase in SRABP with low alcohol, a slight decrease with moderate alcohol. So that stage 2 experiments actually pose so many questions for us to see what actually caused that variations in the lipid levels when we conducted the studies in rats. And we noticed that AMPK and SIRT1 are regulated, and also LKB1 is also regulated. So we wanted to see whether LKB1, AMPK, and SIRT are regulating each other, whether the changes in the lipid levels are dependent on all these three molecules, or whether it is independent of all these three molecules or dependent on only one of those three molecules. So many questions came into our minds, and then we proceeded further with LKB1 role to find out its role in HeLa cells. And when we looked at HeLa cells, so that was something fascinating, because HeLa cells do not express LKB1. So by default, they don't have the LKB1 protein. So it's almost like genetic deletion of LKB1 in those cell lines. So we took those cells and then tried to find out the same AMPK as well as SREBP and SIRT expressions in those cell lines to see whether we can still get similar results as what we have seen with the HepG2 cells and in liver tissues. So in the absence of LKB, if we can get all those things, then we can say that LKB1 is not required. Or if we don't see all those things, then we can say that these effects are LKB dependent. So we proceeded with HeLa cells, again starting with measurements of triglycerides and cholesterol. In every experiment, we proceeded with fresh measurements of cholesterol and triglycerides before we proceeded with the signaling molecules. So we see that a significant increase in free fatty acids, a significant increase in triglycerides in the fatty acid control and cholesterol in the fatty acid control, and then no significant change in both triglycerides as well as in cholesterol with both low and moderate alcohol treatments. Then we went on to look at AMPK expression in the absence of LKB1. So we see that in the fatty acid controls, we see a decline in AMPK and also a decline in phospholipid AMPK. Whereas with the low and moderate alcohol, you don't see a significant change in both AMPK as well as phospho AMPK. Similarly, we looked at SIRT expression. SIRT expression is downregulated in the fatty acid controls, whereas in low and moderate alcohol, there is no significant change. The ACC protein expression is increased with fatty acid control. Its phosphorylation is decreased. So this was consistent in all these experiments. Whereas with low and moderate alcohol, the ACC protein was not significantly changed, even though there is a slight tendency for downregulation in HeLa cells. With SREBP, again, we see an increased SREBP with free fatty acid treatment and no significant change with low and moderate alcohol. So those stage 3 experiments confirmed that the LKB molecule is one of the master regulators on top of AMPK and SIRT1. So the presence of LKB1 is required to show any positive effects. Then we moved on to the next stage where we need to find out the role of AMPK. And we know that there are some AMPK inhibitory molecules available on market. We took one of those compounds called compound C to inhibit AMPK in those cells. We took HepG2 cells for those experiments. We inhibited AMPK expression using compound C, and then we started looking at the expression of other molecules. The cholesterol and triglyceride levels, you can see here in HepG2 cells treated with AMPK inhibitor. In the fatty acid control, there is a significant increase in cholesterol and triglycerides, same as literature findings. In the low and moderate alcohol treatment, you see the triglycerides and cholesterol levels. The triglycerides are increased in both low and moderate, whereas cholesterol, there is a slight increase, but there is no significant difference. Then we looked at LKB1 expression in these cells, in HepG2 cells treated with AMPK inhibitor. So the AMPK expression is inhibited. So we looked at LKB expression. LKB is downregulated in the fatty acid controls, both its protein and its phosphoprotein as well. And in the low and moderate treatments, in the moderate treatment, in fact, you got a significant increase in LKB protein expression. We also see a slight increase in phosphorylated LKB, although there is no significant difference. So that indicates that even though the AMPK is inhibited, LKB expression is still active. So that shows that light and moderate alcohol is regulating LKB expression independent of AMPK molecule. And we also know that LKB1 is actually sitting upstream of AMPK. So there is no role of AMPK in inhibiting or in regulating LKB1. So then we looked at SIRT expression after inhibiting AMPK in HepG2 cells. What we noticed here is a downregulation of SIRT in the fatty acid treated cells. Then with low and moderate alcohol, an increased SIRT expression. So SIRT expression is increased with low and moderate alcohol, even though AMPK is inhibited. So that indicates that AMPK is not required for SIRT regulation. So SIRT1 is regulated by low and moderate alcohol, independent of AMPK. Then we looked at the downstream targets of AMPK molecules, ACC, and also we looked at SREBP. So with ACC, you can see here the fatty acid treatment increased ACC protein, whereas with the light and moderate, with the low alcohol, there is no significant difference. with moderate alcohol, there's a slight decrease and achieved significance in the ACC protein, but not in phospho ACC protein. Then we looked at SRABP in HepG2 cells treated with AMPK inhibitor. SRABP is significantly increased in the fatty acid negative control, whereas with moderate alcohol, we see a significant reduction in SRABP1. So that indicates that SRABP1 is downregulated with moderate alcohol, independent of AMPK, but we know that SIRT1 is active. It's been regulated by alcohol. So that shows that this SRABP1 downregulation is due to or dependent on SIRT1 expression. Then we went on to conduct a similar study where we can inhibit SIRT1. So we have molecules. We use nicotinamide in this particular stage of the project to inhibit SIRT1 to see what its role on the light and moderate alcohol mediated effects. So triglycerides and cholesterol was again measured. So we see an increase in triglycerides, increase in cholesterol in the fatty acid controls, and then with both light and moderate alcohol. So there is no significant increase in triglycerides and total cholesterol as compared with the fatty acid control. Then we looked at AMPK expression, and here we have inhibited SIRT molecule using nicotinamide. So you see AMPK is downregulated in the fatty acid control group, both unphosphorylated and phosphorylated AMPK. And then with the low and moderate alcohol, an increased AMPK and increased phospho-AMPK. So that shows that the AMPK expression is enhanced. It is upregulated by both low and moderate alcohol, regardless of SIRT inhibition. And that makes sense because we know that the LKB molecule, you can see here, LKB1 is increased both its unphosphorylated and the phosphorylated molecule protein. So that shows that LKB1 can regulate AMPK independent of SIRT. So SIRT involvement is not there. So that confirms that SIRT and AMPK, they are actually two regulatory molecules at the same level downstream of LKB1. Then we looked at the ACC protein in these cells treated with SIRT inhibitor nicotinamide. So we noticed an increased ACC in the fatty acid treated animals, fatty acid treated cells, and then a significant reduction in ACC protein expression with the moderate alcohol, but no significant change with low alcohol treatment. Similarly, the SREBP protein expression is also significantly reduced as compared with the fatty acid control group. So collectively, putting all these stories together, what we say is light and moderate alcohol. So we know that we got some significant results with one with low alcohol in some studies, with moderate alcohol in some studies. So we can generalize to say that low to moderate alcohol decrease serum glucose, HDL cholesterol, liver ALT and AST enzymes in the high fat induced fatty liver disease. That's consistent with the literature findings. Long-term treatment with high-fat diet upregulated SREBP, ACC protein, and that contributes to the fatty acid synthesis. That's also consistent with the literature findings. Low to moderate alcohol groups, they show elevated AMPK as well as phospho AMPK, LKB, phospho LKB, and SIRT proteins. LKB1 is essential for the activation of AMPK and SIRT, as demonstrated in studies where LKB1 is inhibited in HeLa cells. Increased SIRT protein levels despite inhibition of AMPK in the AMPK inhibition studies. SIRT deacylates and inhibits SIRBP1C activity, resulting in downregulation of lipogenic SIRBP1C. So that leads to reduced de novo lipogenesis. Low and moderate alcohol increased AMPK via LKB1 in the absence of SIRT, which was again associated with decreased expression of SREBP and ACC. So LKB1 is important to regulate the downstream targets, whereas inhibition of AMPK or SIRT, inhibition of one of them is still is able to produce beneficial effects. That's what we would like to confirm at the end through these studies. And also the threshold of alcohol, for example, the dosage as said in the guidelines, 10 grams, 20 grams for low and moderate respectively. So that's again the borderline or the threshold. That's very much important. That can cause variations in lots of results, because a low alcohol in one particular rat can become a moderate alcohol in a different rat. That's because of the individual variation. So the threshold of the alcohol consumption is also important. Sometimes it is unavoidable when we are at the borderline. With that, I would like to show my past members as well as my present members. So these were my previous lab members, some of my past PhD students and master students. So this person here, Vamsi, so he was the one actually did a majority of these experiments along with Hardik, and he's one of my friends, Dr. Ramesh, principal of Vishnu Group of Colleges in Hyderabad, and Soumya, she finished her PhD as well on a different project, osteoarthritis. So at the moment, so Hardik, he went to Europe and then he came back to India. He got a job as a postdoc in BHU. I heard he's moving to Dubai very soon. Vamsi is still in Australia. And in fact, he got his permanent residency when he applied for Distinguished Talent Scheme with all his PhD results. And then he wanted to propose some new ideas to the government based on new alcohol-based medicated food items. So these are some of my funding agencies over the last many years. I've received significant funding from the Australian government, as well as received some significant funding in the past from Indian government, from the DAD agency in Germany, and also from the universities where I worked in the past and the present. One of our collaborative research centers, NICM, very active in funding and also collaborations on herbal medicine research. And New Colombo plan, that's not for research, that's for teaching collaboration between universities. We are sending some of our students to Singapore. Of course, due to COVID, we can't do it as face-to-face study for the students, but we are conducting some virtual programs for the students. So in future, we can also send some students to India and Indian students to our university through the New Colombo plan. We received some significant funding to send around 15 to 20 students to Singapore program this year. So with that, I would like to thank everyone for attending this. I'm sorry I'm a bit above the time limit. It's nearly 2 hours. I took 2 hours of time. So thank you very much, and I'm happy to answer any of your questions now.
Received some questions on chat box. You can
Yes,
check
please.
it, sir.
Yes, where are they?
On the screen only, sir. You can minimize your PPT, then you can check.
So shall I stop sharing?
Yes, sir. Yes, sir.
Okay. So where are the questions?
On the
Okay.
screen only. Yes.
Yes, I can see that. So what is the better diet in the case of non-alcoholic fatty liver disease? So diet in the sense to prevent non-alcoholic fatty liver disease, or like it's very simple. In order to prevent non-alcoholic fatty liver disease, we know that fat is important for our body cells, but again, we need to keep it in moderation. So not to go for saturated fats, and also fats, high amount of the fat consumption in our diet. So most of us Indians, we do all kinds of deep fries, and then we take a lot of oily foods. So that's one of the ways how to prevent lipid disorders, including non-alcoholic fatty liver disease. What is the fastest way to cure fatty liver disease? Fastest way to cure fatty liver disease is something to see what stage of fatty liver disease we are in, because it's a huge spectrum of fatty liver disease, starting with steatosis or fatty liver disease, and then it develops into NASH, where the inflammation is predominant, and then fibrosis if not treated at that point, and then leads to cirrhosis. So if it is identified at the beginning of the disease, when it is started to show any lipid accumulation in the liver cells, then of course we can actually reverse that by taking appropriate measures to reduce your lipid levels, triglycerides and cholesterol levels. So there are some synthetic pharmaceutical drugs for reversing cholesterol as well as triglycerides. We have statins, we have other substances as well, PPAR alpha compounds, PPAR gamma agonists. So those substances can be used, and again taking into consideration of those side effects. And traditionally, as you know, in our Indian kitchens, we have all those natural products. So lots of herbal medicines. So then they are known to have lipid lowering properties. Substances like garlic, fenugreek, ginger, so curcumin, turmeric. So most of these, they have all these properties, lipid lowering properties. So fenugreek is good, garlic is good, so they're well established. And turmeric, curcumin is known to have lots of beneficial effects on the body, not only to reduce inflammation but also to control glucose as well as lipid metabolism. So if it is at the beginning of the disease, yes, we have some measures to take. But if it is progressed to the late stage, then we have to see the alternative options. How alcohol metabolism says CYP2 pathway generates free reactive oxygen species, whether the formation of reactive oxygen species is more in heavy alcohol drinking. Yes, so with heavy alcohol consumption, we haven't measured any free radicals in these groups, but from the literature we came to know that heavy alcohol consumption generates more free radicals compared with the respective control groups. And also is ROS responsible for inflammation? Yes, of course. So reactive oxygen species, they're responsible for the generation of pro-inflammatory cytokines. HepG2 and HeLa are which cell lines? Okay, so they're both the liver cells. So the HeLa cells are actually the embryonic cells. The HepG2 cells are the liver cells initially isolated from the liver cancer patients. And which culture medium used? So we used EDM, not EDM. I just need to check that one now with my student, which particular medium he has used.
Okay, thank you, sir, for your nice presentation and clarifying all doubts of our participants. So over to Rajaram, sir.
Thank you.
Thank you so much, Dr. Nami Srinivas, sir, for highlighting and sharing your real-time research experience with all of us. Many of the myths and FAQs that were present before are now clear by now, and it will be of great help to all of our researchers who are doing research on the same platform. Thank you so much, sir, for your contribution.
Thank you. Thank you very much.
May I now request our principal and coordinator of this FDP, Professor B.V. Ravi Kumar sir, to felicitate Dr. Nami Srinivas sir with an e-certificate of appreciation as a token of our affection and respect from entire RIPS community.
Thank you once again, Dr. Nami, for accepting our invitation and sharing your research experience with all of us. I must congratulate you for doing research which is useful for the society. And I know you are a very hardworking student, at the same time brilliant student. I know how hard you were working for your thesis. Keep doing all the good work. And I take this opportunity to present you on behalf of Roland Institute of Pharmaceutical Sciences this e-certificate of appreciation. Please accept it.
Thank you very much. My pleasure.
Okay then, Nami. Bye.
Okay. Thank you very much. Thank you, everyone. Bye-bye.
Dear participants, with this, the second scientific session of today's program has come to an end. We are going to begin the third scientific session of day 9 with our resource person, Dr. Ankit Rochani, with us. Before inviting Dr. Rochani to deliver his eminent talk, it is my privilege to introduce Dr. Ankit Rochani, sir, to all of us. Dr. Rochani has earned his PhD in bio-nanotechnology. He is expertise in nano-drug delivery system and formulation science. Sir has been developing integrating nanomedicine concept for cancer therapy that laid the foundation for the concept of personalized nano-drug delivery. Sir has developed various constructs like nanoparticle, nanofibers, and recently he has developed a protein-based hydrogel system for personalized nanodrug delivery applications. His research interests include drug delivery, nanomedicine, LCMS-based proteomics, metabolomics, computational chemistry, and reposition-based approaches for drug design and development. With this a brief introduction, may I now invite Dr. Rochani sir to kindly join us on board for delivering this talk. Welcome to you, sir.
Hello, how are you?
Yes, sir, we are doing good, sir.
Hello, Dr. Sao, how are you? Thank you for this opportunity. So, shall I start sharing my screen? Is it okay?
You go to share screen option, sir.
Yeah, one second.
Yes, just can. Just
That's
can,
fine.
sir. Make it full screen, sir. Make it full.
Is this okay?
Yes. Continue, sir. Thank you.
Okay. Hello, everyone. I'm Ankit. I'm working as a research fellow and an instructor at Thomas Jefferson University Hospital Pharmacy College, Philadelphia. It's evening over there. For me, it's like early morning. So from my end, it's good evening to you all. So my the topic of talk today is about nano drug delivery strategies using smart and green biomaterials. I just wanted to create some kind of a background before I start my talk. I know like many of you are belonging to various working like belongs to various areas of science. I mean, you work in different areas of science like pharmacology, drug design and developments, and like developing new small molecules and checking its anti-cancer, anti-infective properties, and exploring different areas of science in your pharmaceutical um, in the field of pharmaceutical sciences. Now the thing is that like my training started with the synthetic chemistry, where I used to synthesize small molecules. Later on, I used my knowledge of chemistry and used it into like polymer sciences, where I could actually develop nano formulations and which I can use for delivery of the small molecules, which many of you must be synthesizing in the labs. So I've been actually collaborating with number of hospitals, research organizations, as well as companies, and helping them in developing different types of formulations, which mostly are nanoparticles, but I also work on hydrogels. So what I am going to talk about is like some of the basic principles and rationale behind the development of these nanoparticle systems, and which I use for the drug delivery applications. So I just also want to mention that I also serve as a regional and guest editors for vaccines and polymers, where I also help various academicians and research fellows to publish good science. So if any one of you is interested in submitting manuscripts, do let me know. So this is the place where my lab is. So it's a Jefferson College of Pharmacy, and we are a GLP compliant facility. We work closely with the Jefferson Hospital as well as other academic institutions, where we analyze a lot of plasma samples, human tissue samples, and all the biological matrices for carrying out PKPD as well as metabolomic analysis for investigational new drugs, as well as the drugs that are under treatment to patients, and using our LCMS facility, which I have shown on the left side of the screen. We do have rheology and NMR facility with us, where we use it for checking the viscoelastic properties of various hydrogel systems. We do a lot of like nanoparticle analysis using zeta sizers. We also do have access to SEMs and TEMs over here. which I've not shown, but like we do have it. But my talk is going to be more focused towards the rheology and nanoparticles. So like I've just shown some basic facility that which can be accommodated on this slide. So I'll just jump directly into my talk, which I'm going to speak about. So you see, like the world of nanoparticles and formulations would not exist if we do not have a basic understanding of the chemistries of natural materials like like lipids or proteins and polysaccharides. So you see, like a lot of researchers across the world, they are actually working on understanding the fundamental nature of these biomaterials, which can be molded to get either nanoparticles, fibers, hydrogels, or any other different delivery system that exists. So you see, lipids, the most commonly explored material are cholesterol and like maybe PLGA, but PLGA is more synthetic. It's a semi-synthetic or synthetic material. In polysaccharides, cellulose is most commonly explored, which is like later on derivatized to get hydroxypropyl methyl cellulose or ethyl cellulose or different types of cellulose to increase the solubility and other chemical properties of the cellulose material as such. And on the other end, there are like protein-based systems like albumin, which is the most explored biomaterial for the drug delivery applications. So when I'm explaining my talk and like my strategies of developing nanoparticles and formulations using these materials, I'll be more speaking about the rationale behind why these materials were actually explored for developing nano drug delivery systems. So see, the nanotechnology and the nanoparticles and nanoformulations have evolved enormously since probably around 1970s or 1978. I would say that was like one of the golden eras of nanotechnology, because that's when the actual concepts of how the nanoparticles can be designed and developed were actually came into picture, like liposomes or even other form of drug delivery systems. So what happened was that like during this period of the drug, like during this era, we had like polymeric as well as targeted nanoformulations were like introduced, where the drugs like it's water soluble drugs like doxorubicin was encapsulated in a polymeric shell to get a doxyl liposome to deliver it for the cancer therapy. And Doxil was one of the first nanoparticle formulation that was approved by FDA for the drug delivery applications. Later on, like over a period of time, the knowledge of drug delivery evolved and we came into an era which we are into right now, where we are actually developing different shapes and size of nanoparticle systems, because we have more in-depth understanding of how these materials would actually self-assemble to create different shapes of the nanoparticle systems. The most recent one was about like DNA origami, where the DNA as a material was used for developing nanoparticles. And some of my friends in IIT Delhi and IIT Gandhinagar, they are actually involved in developing these DNA based nanoparticle systems. Then slowly we moved into that like solving a problem that like if you give a drug if you give a nanoparticle delivery system, it is more of a passive targeted system. So you put it into intravenous into the human body, and it would just go wherever it wants to go with the bloodstream. But scientists wanted to have like the drug to or the nanoparticles to go more towards the cancer tissue. So they started developing smart surface tagged nanoparticles so that it can be targeting relatively more towards the cancer cells, because like the cancer cells, they have like high expression of certain receptors which can actually differentiate them from the normal cell conditions. So what if we have a ligand which can actually be tagged on the surface of these nanoparticles, so it becomes like a targeted nanoformulation delivery system. So this is the era we are into, where actually the newer designs and concepts have been like evolving in the literature. And my take was that like we still do not have enough understanding of how these materials can be acceptable by a pharmaceutical community like us, whether these biomaterials that we are talking about, are they safe for injection into the human body. So FDA consider it as like a generally regarded as safe categories. So we have limited information about it, but like we have enough information to say that these nano formulations or nanomaterials can actually be injected into the human body. But what if we can use natural systems? Why to go for a synthetic material if we know that the natural systems could be actually be well accepted by the human bodies? So my talk would actually start from a very classical drug delivery design where I used PLGA, which is a synthetic material, and then I'll show like how I evolved into an era where I wanted to more explore the natural materials and maybe use body's own material for actually developing nano drug delivery designs. So I start my story with in IISc. So when I was I was young, I did my master's in pharmaceutical chemistry. I got an opportunity to work in Indian Institute of Science, where I was introduced to a concept of heat shock protein 90 inhibitors. Well, I'm not a biologist from my work, so I would just like give you a brief idea about like what is happening over here. So the lab was working on understanding the role of heat shock protein 90 in infectious diseases, and I was introduced to this concept that heat shock protein 90 is one of the most important druggable targets in cancer therapy. So back in around 2010, there was a really huge buzz about the translation of heat shock protein 90 inhibitors in the clinical trials by National Cancer Institute, United States. So there were like 17 or 18 clinical trials, and some of them, those clinicians who were actually introducing the drugs targeting this HSP90 were our collaborators. So I came to know much about how these HSP90 inhibitors are actually targeting cancer and why they are so important. So you see, the cancer is like a stress. It's like a stressful, the cancer tumor environment is stressful because of the nutrition stress, proteotoxic stress, hypoxia. It's like a completely turbulent environment in the tumor in the tumor microenvironment is extremely turbulent because of these stress conditions that exist in the cells. So you see, even because of this stress, the cell doesn't die, but in fact, it these stress to its advantage, and it still grows. So basically, what cancer does smartly is like uses the defensive mechanism and adapt itself creates a defensive mechanism and adapt itself to the stress condition, and it proliferates. So we found that heat shock proteins were actually helping these cancer cells to grow, because heat shock proteins are basically chaperones which helps in refolding of the damaged client proteins. So you see on the right side, the client proteins that are responsible for the growth of the cells are listed over here, which are helping them in which helps a cell in carrying out its cell division, energy metabolism, and all other biological functions which are important for cells to grow and live. That's how HSP90 helps the cancer cell to grow. So the easier option was that why don't we just kill the HSP90, like just have a drug molecule bind to an HSP90 protein and damage its inherent structure so that it is not able to carry out its function. So I've just illustrated a small, a simple mechanism by which the HSP90 works. So it exists in open conformation state. So in presence of ATP, this open and the client protein, which is kind of damaged because of the proteotoxic stress in the cancer microenvironment. So because of this stress, the client proteins, they become unfolded. So they become non-functional. With the help of HSP90 and ATP, these client proteins, they refold into a close conformational state, and the client protein is released. And it stabilizes the client protein and protects the client protein, and that's how the cell grows, like the cancer cell grows. Now, all this function happens with the help of ATP. And once the protein gets stabilized, the closed conformational state of the HSP90 just goes back to its open conformational state and asks other damaged client protein to take in, and the cycle continues. So the idea was that if we can inhibit an HSP90 protein by targeting the ATP binding site of this protein, it can actually activate the proteasomal degradation pathway, and that's how you can kill a cancer cell. So, so when I looked into like a lot of literature, these were the two, like you can see the pie chart where like different types of heat shock protein 90 inhibitors that were explored for the cancer therapy, and a 17 AAG was the first HSP90 inhibitor that was entering into the phase 3 clinical trial back in like around 2010 or 11. And NVP AUI 922 was at that time was in phase 2 clinical trial. So the problem with these two drug molecules was that like they were given in the DMSO 2% DMSO solutions, and it was like the formulation was very difficult with these molecules because they were like investigational new drug, and as you know, like when the new drugs are introduced, very less like attention is paid towards the formulation of the drug because very less is known about the stability and how to formulate it. So then so now I will go into like how the nanotechnology could help in delivery of these two drugs. So the picture which you should see over here was one of the hypotheses that we had like when we were actually developing a nano drug delivery system. So what we thought was that like even if the HSP90 could be blocked with the HSP90 inhibitor, we will still need another inhibitor with that is like an mTOR inhibitor that could actually help in potentiating the effect of HSP90. So we thought that like, why don't we actually develop a nanoparticle which can have both HSP90 and mTOR inhibitor encapsulated in a nanoparticle system so that it can provide a proper anti-cancer effect. That's what the hypothesis was. But as you know, like encapsulation of two drugs in a single nanoparticle system is extremely tricky because we were not even knowing what was the stability and how would the drug would interact with the biomaterial when it self-assembles to create a nanoparticle system. So to start with, we went into the literature as usual to see like how the drug delivery system was being used. So as you see in 2019, it was shown that like it was shown that like geldenomycin when combined with when given with magnetic hypothermia. So magnetic hypothermia is a concept where you expose the cancer to a magnetic nanoparticle and apply an alternating magnetic field around it. And when you apply this magnetic field, the magnetic nanoparticle causes heating, a localized heating of the tumor tissue, and it can create a physical heat shock. It can increase the temperatures from like 42 to maybe around 50 or 60 degrees Celsius, and it can actually burn the tissue. So what they found in 2009 was that like when geldenomycin, which is a parent drug of 17-AG, which the geldenomycin never entered into clinical trial, by the way. So 17-AG, when given in combination with magnetic hypothermia, it provides like a synergistic anti-cancer potential in mice. Then in 2009 and 10, they found that like 17-AG can be encapsulated with the PEG DSPE to create polymeric micelles. That was a single drug loaded system. Then there was a theory where there was a study where it showed that like mTOR and paclitaxel and 17-AG, they were co-encapsulated in 2011 and 12 into a triple drug loaded nanoparticle. This is like an evidence to our hypothesis that you will always need a combination of drug to treat cancer with a HSP90 inhibitor because cancer is more dynamic in nature. So even though there is an adaptive response, but there are multiple other pathways that are actually happening to help cancer cell to grow. That's the nature. So you need multiple cocktail of drug encapsulated in the polymeric shell to have a proper anti-cancer property. So this was like evidence to my hypothesis that you will need the dual drug loaded system. Then in 2012 and 13, it was found that you can actually create a nanoparticle tagged with the folate, create nanoparticle tagged with the folic acid on the surface of it to create a surface tagged or cancer targeted drug loaded nanoparticle system. So in 2013 and 15, when I was like going with my hypothesis, like what is the next thing that we should do? So I wanted to combine, like, if we can have like 17 AAG loaded with a polymeric nanoparticle system along with magnetic nanoparticles in it. So it was like combining magnetic hypothermia with heat shock protein 90 inhibition characteristic. That's what the idea was in 2013-15 when I started working with this formulation of the 17 AAG systems and designing the nanoparticle system. So as a start, we started with like 17 AG loaded PLGA magnetic nanoparticle system. So what you can see on the screen is the powder, which the maroon color comes from the drug, and the darkness of this is because of the black color magnetic nanoparticles, which are co-encapsulated in the polymeric shell. So it's like a rough way to show that the magnet can actually attract the powder, but and because there is Fe3O4 nanoparticles co-encapsulated with the drug in the polymeric shell, then you see there is a dispersion, and when you put the dispersion close to a magnet, you can see the particles getting attracted to the magnet. And what we found was that like this nanoparticle dispersion can actually help in killing cancer cells because of 17-AG, that is the drug, which is an HSP90 inhibitor. And also this solution, when exposed to an alternating magnetic field, can actually increase the temperature and also provide a heat, like it can provide a physical heat shock to kill the cancer cells. So the characterization of the nanoparticles. So I said that it was a like it was kind of a extremely vague way of representation. Like there is a powder and it can be attracted with the magnet. So you can see under the electron microscope the spherical nature of these nanoparticles. The particle morphologies can be seen over here, and the sum of the particles were also actually mapped to see like how much particle size was there for these nanoparticles when they were manufactured. So the basic synthesis procedure that we followed was like 17-AG, Fe3O4, and PLGA were solubilized in ethyl acetate, and we used oil in water emulsion oil in water emulsion procedure. Classical, it's a classical procedure for creation of nanoparticle in the PVA, where PVA was used as polyvinyl alcohol was used as a stabilizer for generation of these nanoparticle systems. The average particle size for these nanoparticle systems were like around 200 nanometer. But like we actually developed two different types of formulations to see exactly how the polymeric shell would affect the encapsulation as well as the magnetizing properties of these drug-loaded PLGA magnetic nanoparticle system. So you see in the table, there is 1:1:20 formulation and 1:1:10 formulation, where 1:1 is like 1 part of drug, 1 part of Fe3O4, and 20 parts of polymer. And the other one is like 1 part of drug, 1 part of Fe3O4, and 10 part of polymer. And the table shows the polydispersity index, Z average particle size, and zeta potential for the nanoparticles that were synthesized. And this DLS measurements were actually confirmed with the electron microscopy as well. Now, the SEM would only show you the morphology of the drug. So we wanted to see like how, what is the characteristic, like if you want to see the Fe3O4 particle in the polymeric shell, you would like to use other techniques like X-ray diffraction, spectroscopy, and or you can even use like X-ray photoelectron spectroscopies. So in my case, I used X-ray photoelectron spectroscopy, and I used transmission electron microscopy to see like how the polymer shell is affecting, or whether the first question was whether the nanoparticles like the Fe3O4 is present inside the polymeric shell. That was confirmed using transmission electron microscopy, and how the polymeric shell would affect the characteristic of the magnetizing property was confirmed using X-ray photoelectron spectroscopy. So the slide which is shown on the screen, it is like a basic principle how the electron scattering and the characteristic X-rays are being produced. So what happens is that like when you are actually working with transmission electron microscopy, so when an electron beam is hitting your material, there are like multiple things would happen in that scenario. So just imagine if you are putting your sample on a stage and you're hitting an electron from the top on the material. So what will happen is that the primary electron beam would hit the sample, and it would actually cause the ejection of the secondary electron. That's the simplest thing that could happen, and this secondary electron can be used to map the image of your sample. But in certain cases, you also have like backscattering and all the and also generation of a characteristic X-ray would also happen simultaneously. Now, if your electron microscope, like most of the electron microscope, if they're equipped with a detector that can actually detect these characteristic X-rays, then you can actually map the element that is present in that material. Now, in our case, it was PLGA, Fe3O4, and 17AG that was present in the polymeric shell. So to see that there is a Fe3O4 inside the polymeric shell, we wanted transmission electron microscopy to tell us, as well as we mapped characteristic X-rays to see if the particles that we are seeing is actually Fe or not. So the second slide shows you TEM EDS or EDX. So EDX is energy dispersive X-rays, which is like basically measuring the characteristic X-ray which I was talking about. So what you see on the screen is like the first on the left side. I don't know if you can see my yeah, I can get the cursor over here. So you can see over here, the left side, there is a particle which is a normal drug loaded PLGA particle. You don't see any contrast over here. But on the right side, these two images, they are of like, you can see a polymeric shell. It has drug in it, but you can't see it because drug doesn't create a contrast. But you see the dots that you can see over here, it's like it's a crystalline nature of Fe3O4. You see, Fe3O4 is an inorganic material and it has a crystalline property. So you can see a contrast that comes out because of the Fe3O4 particles. They were encapsulated with the polymeric shell. And these two images were actually giving us an evidence that like, okay, the Fe3O4 is actually getting encapsulated in the polymeric shell. So when the electron beam was being fired to get these images, at the same time, the characteristic X-rays were also coming out. So we simply measured those X-rays and we can see the Fe, K alpha and K beta peaks for the characteristic X-rays that can be seen over here. That this is the TEM EDX experiment to confirm that the dots that you see inside the polymeric shell is actually Fe3O4 and nothing else. So you see, we created two different type of polymeric system, like drug systems, that like 1:1:10 and 1:1:20, as I told you before. So we performed X-ray photoelectron spectroscopy to see like how the polymeric shell would actually affect the encapsulation of the material in the polymeric shell. You see, the polymer concentration is higher in 1:1:10 compared to 1:1:20 in 1:1:20 is higher compared to that of 1:1:10. So the panel that you see over here, the x-axis is the binding energy and y-axis is the intensity. It's an overlay of the X-ray photoelectron spectroscopy. This is a different technique than SEM, so please don't get confused with it over here. So you put a sample on a stage and you fire X-ray on the sample. And what you see over here is like the binding energy of the electrons. And what you can see here is that you can see over here, I have marked Fe, oxygen, nitrogen, carbon. Now, you know, like the drug consists of the 17AG, the structure has nitrogen, and Fe3O4 has Fe in it. So we can actually map Fe and nitrogen to see like how the polymeric shell would affect the encapsulation. So what we found over here is that like the top panel, the purple panel is like the standard Fe. The blue and the slightly orangish panel, these two are the drug samples, that is 1:1:20 and 1:1:10. And the bottom panel is the standard drug. So standard drug has like a very good nitrogen signal, which is like not visible in the full length scan. But if we zoom it over here, we can see the nitrogen signal is extremely higher for the 17AG, that is the drug that is green colored. And the PLGA drug-loaded system, 1:1:10, had slightly lower signal, probably because of the polymeric covering. And the red is like the signal completely disappears because the polymeric shell is extremely thick in case of 1:1:20, which is also visible in case of the Fe3O4. Now, this experiment showed us that probably the polymeric shell is actually causing it to become more thicker and thicker. So we wanted to see the drug release characteristic of it, which was also showing us the same thing. So we used UV visible spectroscopy to see how the drug would release from the polymeric system. So you can see the cumulative drug release the percentage drug release characteristic in both the pH conditions, which showed that when you put the nanoparticle dispersion in PBS and two different pH conditions, we could see both time dependent as well as pH dependent drug release systems with the PLGA nanoparticles. Now, since we know that from the XPS that the polymeric shell is actually affecting the signals from Fe and also affecting the Fe signals, we wanted to see the magnetic strength of these nanoparticles using vibrating spin magnetometer. We found that the magnetic strength of 1:1:20 was slightly lesser compared to that of 1:1:10, which is actually suggesting data from XPS that the polymeric shell does actually affect the signals from Fe is because of the polymeric system, because it's becoming more thicker in case of 1:1:20 system. So since the magnetic strength is affecting, so we thought that maybe this could also affect the magnetic hyperthermia experiment that we were actually planning to conduct with these nanoparticles. So as a classical route, we actually wanted to see first the cytotoxicity profile of these nanoparticle formulations in MIAPACA2 cell line. And we found that the drug-loaded drug PLGA MNP provided significant cytotoxicity potential when exposed to MIAPACA2 cell line in time as well as concentration dependent manner. So, and this is because of the drug that is present in the nanoparticle system. Now, the important part of this was also to check whether the magnetic hyperthermia would work in this system or not, because it has Fe3O4 into it. So we took the cells in the tubes. There is a schematic which is shown over here. So there was a circulating water bath where the cells were dispersed in these tubes. And one of the tube was like an untreated system where there was no nanoparticles. There were only cells in it. And the two tubes consisted of 1:1:10 and 1:1:20 formulations. And then we exposed it to an alternating magnetic field. I'm saying this that this was not a heating coil that you see it in India. This is a magnetic field generator. It doesn't cause temperature rise in the system. It doesn't heat up the tube. It just creates a magnetic field. And because of this magnetic field, the nanoparticle resonates to create heating in the solution. Okay, it's not an induction coil which you use for heating your water when you're going for a bath. So although it looks like that, so I'm just suggesting that it's not a induction coil. It is called as a magnetic hyperthermia magnetic field generator. So when you expose it to an alternating magnetic field, it heats up the solution as well as the cells that are being dispersed in the tube, and it killed the cells when we actually checked it using the trypan blue assay in the plate reader. And there was significant reduction as expected, like 1:1:10 was having significantly higher cell death that was seen compared to that of 1:1:20, whereas the untreated system was lying perfectly fine in the circulating water bath, which was exposed simultaneously with the other tested system in the alternating magnetic field. Now, this was a classical case where we actually used the single drug-loaded Fe3O4 PLG nanoparticles. So I also went into dual drug-loaded, like the 17 Ag and rapamycin-loaded PLGA magnetic nanoparticle. So the video which I have shown over on the screen is like the dual drug-loaded system. And the system was later on characterized to see the presence of both the drugs using HPLCs. On the right side, you can see the retention time for 17 AG, and we used UV visible spectrometer with 334 nanometer as a detection wavelength for 17 AG. Whereas, like on the right side, it is rapamycin, where the retention time is 6.1 minute, and they both are resolved they resolve very well on the HPLC system to see the presence of the drug system. And we also checked the SEM images of these nanoparticles, and the average size was like 234 nanometer, and the percentage of the drug was close to 50-50, like for the both 17 AAG and rapamycin in this dual drug loaded magnetic nanoparticle system. Now, moving more into this world of because being a material chemist, my lab's agenda was like to understand how these materials can be synthesized for solving the real time problems. Like, there is one thing that you want to develop beautiful science and develop like really great designs with the nanoparticle, but there is also something called as you also want to develop a simple design that can be which can be translatable for a clinical application. Now you see, cancer is like a very notorious disorder. There was a concept, there was like some concept that was developing at during that time, was that the cancer tumor cells also has a subpopulation of cancer stem cells. So one of my colleague actually started working. We started working on understanding how these cancer stem cells are actually behaving and how they can actually cause resistance to the conventional chemotherapy options. So she actually developed curcumin and GAN61, which is like one of the self-renewal pathway inhibitor. So she developed a dual drug-loaded PLG nanoparticle system for the treatment of which can kill normal tumor cell as well as the cancer stem cells. So what you see is a schematic of a dual drug-loaded PLGA nanoparticle system, which consisted of curcumin and GAN61, and it was used for controlled delivery of curcumin and GAN61 for the treatment of this cancer. On the right side, it is um, the SEM images of the nanoparticles that were synthesized. Now, this was not having Fe3O4 in it. We were trying to understand how could we encapsulate another investigational new drug with curcumin, which is a natural product. So now I'm slowly moving into the world of experimenting more with the natural systems that can be used for the treatment, which are more cheaper to develop and easy to synthesize compared to a complex nanoparticle delivery system. So I'm moved into more into a protein-based system, because you see, like, human body is made up of like polysaccharides and lipids, and the classical example for a lipid being translated into lipid-based system being translated for nanotechnology was doxil, where you develop liposomes with the phospholipid system. So, and the other one is the polysaccharide, but albumin is a natural drug carrier. It exists in the human body, like it covers nearly 60% of the human blood protein. And as a pharmacy students, we all know that the importance of drug binding and free drug and bound form of the drug kinetics that plays an important role in the pharmacological effect of the drug that is being injected into the human body. So I started studying more into this albumin system, and the more I read, I became more extremely fascinated with the system, because it's a natural drug carrier. It exists to carry drugs, metabolites, waste material, everything. It's like a buffer that exists into the human body and creates a sync condition to take material from one compartment to a different compartment. And it was so smart of an Indian scientist who developed this nanoparticle, which was later on approved by FDA and now being used in various cancer therapies, Ebrexane, which is a paclitaxel albumin-bound nanoparticle system, which was one of my inspirations to go more deeper into understanding the chemistry of albumin systems. It was approved like nearly 16 years ago, and even now, Ebrexane is one of the most explored formulations or the nano drug, I would say nano drug, in clinical trials for in combinations with other drug therapies in different type of cancers. So since I was working more on developing therapies for delivery of heat shock protein 90 inhibitors, so I use the background of albumin to create an denatured albumin drug loaded nanoparticle system where luminescent, which is NVP-AUI 922. So if you remember my previous slide where I showed that there were two drugs that were actually more into the field, where National Cancer Institute gave 17 AAG, which I developed with the PLGA, and another drug was luminescent, which was introduced by United Kingdom for HSP90 inhibition. So I used luminescent and developed BSA nanoparticles out of it, and we explored it for the drug delivery purposes and checking its anti-cancer potential. Now, this is the first example of use of a natural material for the drug delivery application, where we used classical desolvation method, or the desolvation method for creating the nanoparticles. So the size of the nanoparticles were in the range of 0 to 500 nanometers, and the zeta potential was negative, which shows that it is stable. The SEM images showed the circular morphology of these nanoparticles, and they would so unlike PLGA where I used Fe3O4, in this case I just wanted to understand how BSA would encapsulate a drug. And then since the drug was getting interacted with albumin, and albumin is a protein, so I wanted to understand the binding kinetics of albumin with the luminescent when it forms nanoparticle. So you can see on the figure which I've shown over here is the fluorescence quenching experiment that was used to see like how increasing the concentration of the drug causes quenching in the fluorescence spectra of the albumin when it starts forming the nanoparticles. Now this is this is something which I wanted to share with you guys. Like when you look at an albumin system, and when you also read the literature, the drug actually interacts with the albumin, and it's a solution. So when you put the drug into albumin, it interacts with the albumin, and that's why the fluorescent quenching happens. But it also indicates that the protein molecules can also aggregate if it goes beyond certain point and form nanoparticles. That's what we observed over here. So albumin on its own works as a nanoparticle system, as a nanocarrier system. But when you go beyond certain point, it can actually aggregate to create a self-assembled nanoparticle system, which can be seen visibly, and it's a dispersion. So what you see over here is like increasing the concentration causes reduction in the fluorescence quenching, which causes the drug to quench, like fluorescence to quench, which indicates that the albumin interacts with the drug. And when the nanoparticle synthesis is complete, the fluorescence quenching is extremely low. Like it just drops extremely low compared to a normal BSA nanoparticle. So which is also indication that the drug interacts with BSA when it forms nanoparticle. Whereas normal BSA nanoparticle has significantly high fluorescence intensity, which is shown in blue color, which was which showed that the drug interacts with the albumin. We also confirmed that using the UV visible spectroscopy, where the green line is the standard UV visible absorption maximum for the NVP-AUI 922 or luminescent. So luminescent can be given by two names, like NVP-AUI 922 or luminescent. And the red line is the drug loaded BSA nanoparticle, and then the blue line is like a normal normal BSA nanoparticles. So since the drug was interacting with the BSA, we wanted to see like where it would go and interact. So we could see the binding. We performed a docking study to see like where the drug would go and interact to form the nanoparticle. So this is a three-dimensional model. The Gibbs free binding energy was minus 10.48 kilocalorie per mole. And there were like a lot of hydrogen bond and polar interaction, which shows a stable structure when it goes and binds to the albumin binding pocket when it forms the nanoparticle. Then we wanted to see like how it would go and affect the whether it has an anti-cancer potential or not. it did have an anti-cancer potential time as well as concentration dependent manner for both MIA-PACA and MCF7 cancer cell line. On the right side, so you see, like in this assay we use LMR blue assay to map the cytotoxicity potential. But like it's also important to visibly see exactly how the cancer, like how the cells are being damaged, because when you expose it to the drug loaded system. So the top panel is like the MCF7 cell line when we added BS nanoparticles. So you can see there's a little bit of debris that is just floating around, and the colonies of the MCF7 cell lines just being formed. But when you actually expose it to luminescent, you can see like the morphology becoming shrinked, and the cell starts to die when you expose it to luminescent loaded BSA nanoparticle, which is like a qualitative way of actually suggesting that yes, the nanoparticles do help in having the anti-cancer potential. So later on, like moving further in the world of nanoparticles, we have like drug-loaded neosomes that were developed, where cholesterol, the lipid-based system, was used for delivery of another natural material, morocin, for anti-cancer drug therapy, where we use like thin film evaporation method for the drug delivery application. The right side, these are the SEM images and the particle size distribution characteristics and of these nanoparticle systems. We also developed nanofibers using cellulose. So cellulose is water insoluble. So we created water soluble cellulose by acidosulfation method, and bacterial cellulose were sulfated, and these sulfated bacterial cellulose were actually water soluble, and we could actually develop nanofibers using them using electrospinning method, and we could see the cytocompatibility of these cellulose nanofibers on L929 cell lines, and the cell lines could actually grow over these nanofibers, and it was like working wonders. And it shows the cytocompatibility properties of these nanofiber systems. Okay, so now like these nanofibers and nanoparticles, like it's really good to manufacture in the lab scale. Nanofibers are really like little tricky because you need an expensive equipment like electrospinning machine to actually form these nanofibers. So since nanofibers were like tricky and little expensive, I've ventured more into hydrogel system, which is relatively easy and simple to make. So to go into the hydrogel, I've just shown on the slide where you can characterize a Newtonian, non-Newtonian system, where Newtonian systems, they like when you apply shear stress versus rate, the Newtonian system are linear. Pseudoplastic systems are shown in blue, and all other non-Newtonian systems are shown in green, Bingham, and plastic and dilatant systems. Like these are like the classical way of expressing how a hydrogel would work. So the reason I'm showing you this is because like this is like a classical way of mapping how a hydrogel system would work, because they are viscoelastic in nature. So when you see a Newtonian system and you perform a rheology for it, on the left side, this is a Newtonian silicon oil. The viscosity is in linearity. So the green line is like the green line is a stress versus shear rate. It's linear, and it's a non-Newtonian, which is like same as that of what I've shown it over here. And when you take a hydrogel, it's a pseudoplastic system, like it goes like this. And you can see over here, pseudoplastic means a shear thinning system. Sorry, the shear thinning system. Now, this is the classical difference between a Newtonian and a pseudoplastic system. So most of the hydrogels that we synthesized, they were like pseudoplastic in nature. And we are going more into something which is called as physical and chemically cross-linked systems. And we will be talking more about something called as a storage modulus and loss modulus. And to simplify that, storage modulus is the property of a material to store energy. That means it is more elastic, and loss modulus defines as the material to dissipate energy, which is like a liquid nature. And most hydrogels, when you perform a rheological experiment, you can see a spin Barkite behavior of the gel system. So there is a difference between a viscous, viscoelastic, and an elastic system. So when you are actually performing these rheologies, you can actually differentiate between viscous and viscoelastic and elastic systems. Now the gels can be chemically as well as physically cross-linked. The character, like when you perform a frequency sweep experiment where you measure the storage and loss modulus versus angular frequency, the physical cross-link system, they don't cross each other. That means the storage modulus will be always be higher than that of the loss modulus. Whereas in case of a chemically cross-linked system, the storage modulus is initially lesser, but it then later on crosses the loss modulus, giving you a crossover modulus over here. So just to simplify it and make it easy for you to understand, this is a characteristic of a chemically cross-linked system, and this is a characteristic of a physically cross-linked system when you perform a frequency sweep experiment. Most of the natural systems that exist, to my knowledge, they are physically cross-linked systems. So the when you perform rheological measurements with these carrageenans or chitosan or albumin or any other hydrogel system that exists, most of them are physically cross-linked system because they get gelled when exposed to a physical stimulus. Instead of have a chemically cross-linking reaction, it has more of a physical cross-linking that is playing role in formation of the gel. So what you see over here is because in the initial experiment we saw that there is a physically cross-linked system, we also wanted to see if a tissue, whether it is a physically cross-linked or chemically cross-linked, because that would make sense that like most of the system, why they are actually physically cross-linked instead of chemically cross-linked. So the brain tissue, when exposed to rheology and we performed a frequency sweep experiment, we could see that even the brain is first of all, it is viscoelastic, which we could see from the images on the rheometer like this. The stage has the brain tissue from a mice. You crush it, you perform the rheology, and it shows the spin bicard behavior same as that of a hydrogel, which shows that it is viscoelastic in nature. And then when you perform a frequency sweep, it shows that the storage modulus and loss modulus never cross each other, which is a characteristic of a physical system. And on top of it, because it is viscoelastic, we could actually map the viscosity. It is a pseudoplastic system, which could be seen from the shear stress versus rate experiment. Now, when we were actually making our hydrogel, we wanted to see like whether these hydrogels could be matching to the tissue system where we want to put it, because we are planning to develop an injectable hydrogel system which could be injected into the brain tissue for localized drug delivery, so that like the gel would actually mimic the localized tissue area. So the panel which you see over here, it's like water. It forms like a harder gel. You need 25 millimolar sodium chloride to form a transparent hydrogel with the albumin. And as you increase the concentration of the material, the gel becomes harder and it becomes more structured when you form a 10% gel. And from the frequency sweep experiments, we could see like the albumin also is a physical system where you have the storage modulus is higher than the loss modulus. And we used, because it's a physical system, we used a thermal denaturation method where the protein get denatured and it aggregates to form a transparent hydrogel. So when we expose it to heating at 85 degrees Celsius at like 30 minutes, 45 minutes, and 60 minutes, this experiment on the right side, it's a thixotropy measurement. And as the temperature increases, the system becomes more harder and harder. And it shows the formation or the transition of a solution to a viscous, to a gel, and extremely elastic system. So you can actually, by monitoring the time of exposure and the heating of the system, you can actually manage the thickness or the thinness of the material that you want to, or the drug delivery system that you want to develop. So we loaded drug with this gel. So we loaded doxorubicin into it because the doxorubicin is water soluble, and it's like a classical model drug that can be explored. So we use this as a model drug for checking its loading characteristic and its viscoelastic behavior. We again performed the frequency sweep at 25 degrees Celsius as well as 37 degrees Celsius in this case, and we found that there was like negligible difference that exists in like there is a difference in like 25 versus 37 degrees exposure, but it's just slightly lesser. But my experience says that there is a difference which exists in the 25 and 37 degrees Celsius exposure of the gel. So when you see the body temperature is 37, so when you expose these gels to the 37 degrees Celsius, there is a slight variation in the viscoelastic behavior of the gel when you inject it into the human body. So to check whether these gels could actually kill cancer cells, we exposed it to MCF7 cell line. So you can see here the 1000 nanogram so per ml concentration of the gel. So the gels were actually broken down into pieces and they created a dispersion out of it. And these dispersed gel pieces were then exposed to the cells. And you can see the cells were like slightly they were growing, but they were like the concentration of the cells growing at 1000 nanogram per ml was slightly lesser, whereas with the increase in the concentration to 5000 nanogram per ml of the gel piece was giving more killing of the cancer cells. So having like with the understanding of the albumin causing the formation of the gel, and it can also be used for the drug loading characteristic, and we know that the albumin is a physically cross-linked system, and we also knew that from our with the multiple experiments with the students in my lab, that materials like polysaccharides like carrageenan or chitosans, they also are physically cross-linked system. We wanted to develop some system that could be physically cross-linked at the acidic pH, but when you increase the pH, it becomes chemically cross-linked. So this is the classical example of that system where we mixed PVA boric acid, where it was so PVA when PVA reacts with boric acid in the basic pH condition, and it forms a boronic cross-linking. And what we did was we mixed PVA and boric acid with carrageenan to create a composite mixture. And at acidic pH, they behaved as a physically cross-linked system. But when we increase the pH of this PVA boric acid carrageenan to pH of something around 7.5 to 8.5, which is shown over here. So it was initially it was like liquidy. When you increase the pH, it becomes like a chemically cross-linked system. And this system we are actually exploring to create female contraception, because the uterine pH, the cervical mucus pH is like around 4.5. And we think that when the seminal fluid interacts with the cervical fluid during the intercourse, it can increase the pH of that situation. And if we can mimic that situation, then we can actually put these gel in the cervical mucus, and it can serve as a physical blocking agent, and a contraception can be developed using this gel system. Now, apart from the gels and the natural systems that are explored in my lab, we also do a lot of mass spec based work where we help clinicians in investigating the clinical trials, the ongoing clinical trials in Jefferson Hospital. So the slide that I have shown over here is like all the different types of projects that are going on in the lab, where we do a lot of metabolomics and PKPD analysis of different types of drug molecules that are being investigated in the patients. And we develop a highly sensitive LCMS method in our lab. It's a GLP compliant lab, so it just gets audited almost every time by different regulators, different clients. And these are some of the book chapters that I have written for some uh, for eminent publishing houses. Like you can look at the published literatures regarding the natural materials that are explored for drug delivery, if you are interested, or the role of each of protein 90 in infectious diseases or cancer. You can check. Slow down. This is also a guest issue that I am leading in polymers. So it's a 3.5 impact factor journal. So if the faculties and principal investigators, if you're interested in submitting manuscripts for nano drug deliveries and formulations, please do get in touch with me. So this is my lab and this is my PI, and these are all the students that we have in the lab. And most of this work is supported, some of this work was done in my previous lab in Japan. So it was supported by MEXT and BioNano Center in Toyo University. And most of the work right now is being funded by R01 grant in NIH from NIH as well as from the Jefferson Hospital. And the students, if you are listening, if you are interested in joining the MS in Pharmaceutical Sciences program in our institute, sorry, I'm taking this chance to propagate this information, but if you are interested to join in MS in Pharmaceutical Sciences, please get in touch with the department head, Dr. Alok Bhushan or Christine Lord, which is shown in the slide. You can even give a Google search, you will find this page, MS in Pharmaceutical Sciences, Jefferson College of Pharmacy. And they are giving GRE waiver for your information. So you don't need to give a GRE examination for getting admitted for this course. And it is for short time only. So I am not sure what will happen next year. So right now this is going on. So I just wanted to inform you all. And you see, I've been working on different types of gel systems. So like what I would like to show is that the albumin can serve as like an injectable gellable systems. It can also be it can also form a structured transparent gel system, which is more elastic and less viscoelastic. And we also molded these albumins to print these Jeff. Jeff is like a short form for Jefferson. So it was like some kind of a fun activity that was going on in the lab. So we just printed this Jeff using albumin solutions. So I think that albumin works really good. Thank you so much for your attention and thank you for this opportunity. I'll be happy to take any questions that you may have.
Thank you, sir, for sharing your research experience. So definitely collaborate any research of our faculty or any research scholars, and those are interested for any publications. Research also can coordinate with you, sir. Thank you, sir, once again.
Sure, no problem. I'll be happy
Over to Radharam, sir.
Yeah, thank you so much.
Thank you, sir.
Thank you so much, Dr. Ankit sir, for delivering a talk on such an innovative drug delivery system. Most of the systems are nowadays going nano, and we hope that this NDDS is going to be the future of healthcare facilities very soon. We remain most grateful to you, sir, for the rapt attention with which you have delivered this presentation. Thank you so much, sir. May I now request our principal and coordinator of this faculty development program, Professor B.V.V. Ravikumar sir, to felicitate Dr. Ankit sir with an e-certificate of appreciation as a token of our affection and respect from entire RICS community.
Thank you, Dr. Ankit, for accepting our invitation and sharing valuable information about the use of green biomaterials in nano drug delivery systems. We are grateful to you. And on behalf of Roland Institute of Pharmaceutical Sciences, I would like to present this e-certificate of appreciation. Please accept it, sir.
Thank you so much, sir.
Thank you, sir.
Thank you so much.
Thank you so much.
Thank you, sir.
Thank you so much, sir. No problem.
So dear participants, with this, the day 9 of FDP has come to an end. We'll be sending the test link for this one, test link for series 3 through your registered email ID. For this ongoing COVID situation, we are very sorry to inform that this valedictory function could not be held tomorrow at the scheduled time. But we are going to select this valedictory function. We're going to happen this valedictory function very soon, and we'll intimate you through your registered email ID regarding the date and time also. So till then, please stay safe and stay connected with us. Thank you all the participants for your active participation and coordination with us. Thank you very much.